computer. Okay, does it tell me when I'm recording? Okay, pause recording. I'm assuming I'm recording. No. Ben, there we go. All right, that's who we're missing. All right, so here we go. On these notes, so cells are going to divide for several different reasons. We're going to talk kind of out of order of these. For growth, mainly that's going to be the big one. You're growing still. Plants grow as well. If you're a living organism, you grow. That's one of the tenets that makes something alive, is to actually undergo growth. Single-celled organisms, prokaryotic, eukaryotic, they all grow. And so this is going to be the growth of an organism. Now, the cells don't necessarily grow in size. If they do get a little bit bigger, what eventually they'll do is they will split in half, and you will have what we call identical daughter cells. That's mitosis. Okay. You also do this with repair. You repair damaged epithelial tissue. That's your skin. So let's say you get a paper cut. It's going to form a clot so that you don't bleed to death from this paper cut. And then what's eventually going to form is new skin underneath the scab. So it's going to scab over, and then you get new skin underneath the scab, and eventually the scab will fall off. And it's magic. Well, it's not magic, it's mitosis, right? So these cells around it, these epithelial cells, are going to undergo mitosis, growth. They're going to split and replicate, making identical cells, identical copies of themselves. And then they're going to reform the skin that you had. You do this constantly when you're eating, because your stomach, being pH of 2, is constantly digesting itself. And so you are undergoing a really high rate of mitosis in the lining of your stomach. If you were single celled organisms, you would be doing mitosis for reproduction purposes. We are not. So we don't do mitosis for reproduction. We do meiosis, which is a different process we'll talk about after winter break. If you are a single celled organism undergoing the process of mitosis, we call this asexual reproduction, meaning you don't need a partner in order to reproduce. You just grow and replicate yourself. It's essentially like cloning. You're just going to create an identical copy of yourself. Specifically, we call this binary fission. That's just the fancy term for mitosis of single-celled organisms: bacteria, yeast, amoebas, parameciums, volvox. It's going to create a daughter cell that is identical to the parent cell. The daughter cells are the new cells, the parent cell is the old cell. And they just split. All right, when you were a tiny little ball of cells, so sperm meets egg, fertilization happens, you're a tiny ball of cells. You are undergoing the highest rate of mitosis you will ever undergo in your entire life. And it's all you do as this tiny little ball of cells is you basically will grow and split, grow and split, grow and split, making identical copies over and over and over again. Eventually, you will make enough of those identical copies of the cell that they will start to differentiate. So when they're undifferentiated, they are not specialized. All your cells are the same. We call those stem cells. Okay. So stem cells are undifferentiated cells. Through the process of mitosis and undergoing cell division over and over and over again, you will specialize those cells by either tightening down or loosening up certain parts of the DNA. We call that essentially turning off or turning on certain genes. That is so that some cells will become your epithelial cells, your skin cells. Some cells will become liver cells. Some cells will become brain cells. They don't look the same, but they all contain the same DNA and they all came from the same source. So they will eventually specialize or differentiate because of the rate of mitosis and where they are in that rate of mitosis. Your bone marrow is a perfect example of where you continue to do cell differentiation. Now, it's not like the cells out of your bone marrow are going to undergo like new cells. They're all going to become bone cells, but they're going to become what we call osteoblasts, osteoclasts, osteocytes. So basically, they're responsible for growing new bone, maintaining the mineral deposits in the bone, or harvesting mineral deposits from your bone. So you do still have cells that differentiate inside your body based on their rate of mitosis and where they come from. Taj, did you have a question? Oh, no, you're not. It's the projector. Yeah, so when the air is on, it vibrates the ceiling, which vibrates what the projector hangs from. Yeah, when they also are moving things upstairs, because this is the physics classroom, when like Dr. Pierce is dropping stuff and moving things, it shakes the projector. Yeah, it's super fun. I'm always like, oh, they're doing this experiment. I can tell by my projector shaking. Somatic cells are going to be body cells. So somas, it's ancient Greek. It literally means body. So these are body cells. We differentiate because you do have cells that are not your body cells. All of your somatic cells have a full complement of your DNA, which means they have 23 pairs or 46 chromosomes. And those are the chromosomes that make you you. Your DNA is what makes you unique. Everyone has their own unique DNA. You share very similar DNA sequences with your family members. Makes sense. Genetic family members. However, you do have cells in your body that do not have a full complement of 46 chromosomes. We call those gametes. And that would be eggs and sperm because we are sexual organisms. We don't do asexual reproduction, we do sexual reproduction. You have to find a partner. So, spermatozoa is a fancy term for sperm. If you're a plant, this is called pollen. Okay, pollen is plant sperm. And then an oocyte or an oocyte, depending on how you want to call it. I call them oocytes. That's just eggs. Eggs and sperms are your gametes. Gametes are going to have half the number of chromosomes, which makes sense because if you find a partner and you have a sperm with half the number of chromosomes and you find an egg with half the number of chromosomes and they fuse together, you'll get an organism that has 23 pairs, one from the sperm, one from the egg, or 46 total chromosomes. Those are called gametes. Everything else is somatic cells. All right, here we go. 90% of the time your cells spend in interphase. Most of their cell life will be spent in interphase. Interphase is where they prepare to divide the nucleus. Mitosis is only the division of the nucleus. You need to understand that prophase, prometaphase, metaphase, anaphase, telophase, that is only the movement of the chromosomes and the splitting of the nucleus. The cytokinesis is the splitting of the entire cytoplasm or the entire cell. G1 is where the cell matures. And this is really going to be where the cell is just collecting nutrients. Does it have enough nutrients to double its number of Golgi bodies? Does it have enough nutrients to make full copies of its DNA? Does it have enough nutrients in order to make the endoplasmic reticulum and the free-floating ribosome and another nuclear envelope? Because when you make a new cell, you've got to make another nuclear envelope to protect that new DNA you've just made, okay? So it has to basically do supply collection. Then it starts to do cytoplasm and organelle duplication. Then it's going to say, okay, yes, we have enough nutrients to go ahead and replicate our DNA. So then it's going to go through and replicate the DNA. That is S phase, S standing for synthesis. Okay. So that's going to be the synthesis of new DNA. That is the S phase. G1 and G2 are growth. And then G2 is going to be the cells preparing for division. So this is going to be where it's going to start growing its plasma membrane, the phospholipid bilayer on the outside, because you don't want to split the cell and have it go back to being like half the size of the parent cell. That wouldn't make sense, especially if you're replacing damaged cells, if you're replacing it with a cell that's half the size. So this is going to be G2, where you're going to do replication of the phospholipid bilayer. You're going to have your cytoplasm split in a way that basically your organelles are moving to opposite sides of the cell, because the cell is going to split down the middle or the equator in cytokinesis, and you want to have the full complement of mitochondria, the full complement of the Golgi bodies, all the things that you need, the ribosomes in each one of the cells. You don't want to have all the doubled organelles end up in one cell and then an empty shell of another cell. That makes no sense. Okay. So that's going to be the G2. And then cytokinesis is when the whole thing splits. M phase is pretty quick. All right. Chromosome. So here we go. I like to call these replicated chromosomes. If it's in the shape of an X like this is a replicated chromosome. This is not how your chromosomes normally appear most of the time in your cells. This is how we see them when they are going to undergo mitosis. But understand, just like in the previous slide, because it'll go back, when it's undergoing mitosis, it's already copied its DNA. It's already replicated all of the chromosomes. So what we see under a microscope when we see the chromosomes move through prophase, metaphase, anaphase, telophase is that they are replicated. That's twice the number than what you need, right? So this is not normally how your chromosomes appear. How your chromosomes normally exist in interphase is as chromatin fiber. So that's going to be the double helix that you know. Hopefully, you know your DNA is a double helix. It's going to be wound around these histones. These are proteins called histones. And then those histones are going to be wound into what we call chromatin fiber. That chromatin fiber is then super looped, and then those loops are wound up and looped again. And this is what we call it's super condensed DNA, right? It only does this because it needs to move it to the opposite side of that nucleus area, the nuclear envelope where it was, so that it can split down the middle. It's much easier to split the nucleus when your DNA is nicely condensed in these super coils and you can just move a chromosome instead of having to drag this like bowl of spaghetti to the other side of the cell. Okay. So this is what we see when they're replicated. It's super packaged like that. This is just called supercoiling. This is a replicated chromosome. Normally you only see one of these. For the chromatin fiber, this is how you turn on and off your DNA, is you tightly coil it around these histones or you loosen it up around the histones. And we'll learn about that later in unit 6 when we do gene regulation. But it's, if you want to know, it's called methylation and acetylation, if you're curious. Okay. So here we go. This is going to be really similar to the idea of like photosystem 1, photosystem 2 kind of thing, how they had the naming convention and then they realized later on because of technology that it wasn't quite right. And so then they went back and they used super confusing names. So here we go. If you see it in an X, that is a chromosome, but it is a replicated chromosome. Okay. It is comprised of 2 chromatids. When it is single by itself, that is also a chromosome. Right? Yeah, see? So it's a 1 chromatid chromosome versus a 2 chromatid chromosome. And you might see them on the AP called unreplicated chromosome versus replicated chromosome. Okay. So if it helps you to be like, it's a 1 chromatid chromosome versus a 2 chromatid chromosome, you can call them that. I'm fine with that. But just understand like where they exist. So this is going to exist before mitosis. So the majority of your cell is spent as this single 1 chromosome, the majority of its life, because this is how it's going to occur in G1 of interphase. This is after S phase of interphase. And then after you undergo anaphase, because remember metaphase, they all line up like X's in the middle. And then anaphase is where they start to pull apart. The 2 chromatids are going to pull apart. That is where you're going to get this 1 single chromatid again after anaphase. So that it's a single chromosome, 1 chromatid chromosome, unreplicated chromosome that's going to go to one side of the cell. another unreplicated chromosome that's going to go to the other side of the cell. And then it's going to reform the nuclear envelope under telophase. And then cytokinesis is going to split the whole cytoplasm. And you get two identical daughter cells that look just like the cell before mitosis started. Okay. The middle region that is always kind of denoted with this like weird circle is called the centromere. That is a region of non-coding DNA. It does not contain a gene. And it's in the middle like that because what it does is it contains kinetochores. And a kinetochore is a region for which these centrioles are going to send those microtubules, and the microtubules are going to attach. And those are what is going to pull the chromosomes to the opposite sides of the cell. It's really cool because you have signals that literally like motor proteins that like walk away that tell the cell that it's time to like pull apart. Yeah. So that's really cool. That's new research. How they know where to pull them. So they used to think that it was like a fishing line kind of thing, right? Like the centrioles like get to the poles and then they send out the microtubules that kind of hit the kinetochore region and they like reel it in across the cell. That's not how it works. It's really cool because they did research where what happened is that so they've done research where you have like the circle, right? And you've got the chromosome in the middle for metaphase, and then here's like little centriole. So they thought it was where it sent out the fishing line kind of thing. It hooked the chromosome and then it pulled it back this direction. But what they found is if they cut the line right here, the chromosome would still move to that point. But see, right, if it were a fishing line and you cut the line, you wouldn't reel in the fish anymore. And so what they realized is that the movement of the chromosome is not coming from the centriole. The microtubule comes from the centriole, but the movement of the chromosome comes from the chromosome itself. And what they do is they send cyclin-dependent kinase. They send kinase signals back and forth, almost kind of like they're pinging, like how sonar works, where they send a signal and then the signal gets sent back and then you kind of know where it is. So the chromosome will basically ping the centriole and then the centriole will send a signal back and then the chromosome knows where to travel. It's amazing. Huh? They don't know that yet. Yeah, Nobel Prize winning stuff. Do you know how the centrioles get to, do the centrioles know where to get, where they go? Right, it is. Oh, that's such a beautiful way to say it. It is. It really is. Intracellular echolocation. They're all dolphins. Your chromosomes are dolphins. Okay, don't put that on the AP, by the way. They'll have no idea what you're talking about. Like, your chromosomes are dolphins. Okay, so interphase. Remember, 90% of the time, this is before S phase because they're unreplicated chromosomes. These are the one chromatid chromosomes. So DNA is long, it's thin, it's chromatin, it's uncondensed. This actually would be like spaghetti. You wouldn't be able to see it. The centrioles are actually not really there. They don't quite know where the centrioles go in interphase, but you can't always see them. And they don't know if they disappear, reform, or if we just don't see them. And then interphase after the S phase is the DNA has replicated into what we call sister chromatids. That's because they're the exact same DNA. They're just attached to each other at that non-coding region, that centromere. And then the centrioles have actually replicated because you need one pair to go to one side of the cell and another pair to go to the other side of the cell. In mitosis, this is where we're going to start, remember, because this is the movement of the nucleus. It's the splitting of just the nucleus. So that nuclear envelope has to disappear. So it gets digested, essentially. It's going to disappear. It's a phospholipid bilayer around your nucleus. And those centrioles are going to migrate to the poles. And the poles are just going to be the opposite ends of the cell. So you can kind of treat it like it's a globe. So this would be like a pole right here and a pole right here. So these are the poles. Need stylus very badly. There we go. So there's a pole right there and there's a pole right here. And just like the globe on the earth, if you have a pole, you have an equator in the middle. So that's the equator. So in prophase, nuclear envelope starts to disappear. DNA starts to supercoil and condense. The centrioles are making their way to the poles. And then in pro-metaphase, this is kind of this weird, like it's after metaphase, so it's after prophase, but it's before metaphase. The centriole pairs are going to like further migrate. The nuclear envelope fully disappears. These are the kinetochore spindle fibers are going to capture the kinetochores on the chromosome. Basically, what that means is the spindle fibers, those microtubules produced by the centrioles, are going to attach to the chromosomes at the centromeres. That's what a kinetochore is. And then those spindle fibers are going to kind of push and pull. So they're doing this like push me, pull you, because what's happening is these chromosomes are attempting to line up in the middle. And you're like, well, how do they know how to get in the middle? And it really is a push me, pull you kind of tug of war between the centrioles at the poles. And then they end up in the middle like this. Okay. And they use tension. They literally use like your chromosomes, your kinetochores use the tension of the spindle fibers to know when they've hit that middle. All right, so the chromosomes are going to line up along what we call the equatorial plate. That's where cytokinesis is going to take place. Your cell will split along the equator. And then in anaphase, now here we go. Metaphase meaning middle. They line up in the middle. These are all those replicated chromosomes lining up their sister chromatids in the middle. The spindle fibers are attached to the kinetochores. In anaphase, there is a checkpoint right before this process happens. Okay, so I'm going to put checkpoint. This is where your body is going to check. Your cell is going to check and say, okay, are all the spindle fibers attached correctly? Are these sister chromatids going to split where we get one sister chromatid in one area of the cell and another one in the other side of the cell? Basically, they want to avoid one whole replicated chromosome going over to one side of the cell and nothing going to the other, because then you're not ending up with identical DNA. If there is something wrong with the attachment of the spindle fiber to the kinetochore, your cell will not undergo anaphase. It will either go back to interphase. So it'll basically go into what we call a G0 holding phase. It will not split, or it will undergo apoptosis, which is programmed cell death. Okay. So it does, your cells do have a checkpoint for proper attachment. So it's not just gonna, this is basically to prevent inadequate DNA and too much DNA in the replicated daughter cells. The kinetochores are then going to shorten, which basically means they kind of like march their way back to the poles right here. These kinetochores shorten. The chromatids are at the opposite ends of the poles. The microtubules kind of push against one another and help elongate the cell. So you notice right here, these microtubules, the ones that aren't attached to chromosomes, actually will lengthen it into an oval shape. And that's fine because in G2, you've already replicated the plasma membrane enough that it can split down the middle. Telophase is where you're going to basically the opposite of prophase. So in prophase, nuclear envelope disappeared. Telophase, you're going to reform the nuclear envelope. In prophase, the chromatin became condensed chromatids. In telophase, the condensed chromatids are going to become uncondensed chromatin. Okay, so it basically, telophase undoes everything that happened during prophase. And then what's going to happen is in an animal cell, you get this dip, and it's called a cleavage furrow. And that's going to be where cytokinesis takes place. And in a plant cell, it's just called a cell plate. It's just where the new cell wall forms in between two plant cells. So your M checkpoint of mitosis is in anaphase. You need to know that. Your other checkpoints are in G1 and G2. You have 3 checkpoints in the cell cycle. There is no checkpoint in the S phase because pretty much if your cell receives the go signal to replicate your DNA, it's going to go ahead and replicate the DNA. It doesn't want to stop in the middle of such a delicate process. All right, anybody need any more time? I'm going to post this, by the way, if you didn't get all of it. I'm going to post like the 2 cells with all the boxes with stuff in it. just so that everybody has stuff to get. All right, so here's the checkpoints. You've got G1 checkpoint, you've got G2 checkpoint, you've got M checkpoint. It says metaphase, but it's basically right in between metaphase and anaphase. It's essentially going to be the checkpoint right before you pull the DNA apart. So the checkpoint's basically going to ensure that the replicated cell is healthy, it has enough nutrients, and its DNA is where it's supposed to be. So in G1, the restriction checkpoint, this is, do we have enough nutrients to go ahead and go? Then if we do, we're going to synthesize the DNA. Then you're going to replicate pretty much everything. You're going to check the DNA for damage. You're going to check that you have replicated all of the proper organelles. And then you're going to go ahead and split the DNA. And right here, you're going to check that the spindle fibers have attached correctly to the kinetochore. And then you're going to go ahead and pull them apart and then split the cell. So you do have 3 checkpoints. Of those 3 checkpoints, your entire cell cycle is what we call cyclin dependent. Cyclin dependent, these are going to be your cyclin signal. So G1, you have to hit a high enough concentration of this cyclin in order to progress to the S phase. And then the S phase you're going to go through and then get another, you're building your cyclin concentrations. And then for G2, you have to have a high enough concentration to send that signal to go ahead and split, and then send that signal to go ahead and split. You're going to be sending those signals to cyclin-dependent kinases. So you have to activate enough of the kinases in order to progress. Lack of the checkpoints or lack of the regulation is going to lead to unregulated cell growth. If you have unregulated cell growth, that can lead, it won't always lead to, but it can lead to tumor growth or cancer. And if eventually that is so much unregulated cell growth that it starts to break off, pieces of your cells start to overcome what we call anchorage dependence, it will then start to spread, which means that it has metastasized and is spreading to other parts of your body. Normally, we refer to that as malignant. It is a malignant cancer or malignant tumor if it is actively growing, overcoming population density, and overcoming anchorage dependent checks, spreading to other parts of your body. Okay. So what is happening? This is a fish blastula. This basically would be where? Can you see any condensed chromosomes? Like kind of, right? But not really. Right? You can kind of see it. You can kind of see an envelope around them. So this might be like transitioning between like interphase, so G2 and like prophase, because there's still that nuclear envelope, and you can kind of see some darker images, but it's still really fuzzy. This one right here, however, you can definitely see these spindle fibers starting to form. Nuclear envelope is a little bit fuzzy over here. You can still see it a little bit more over here. Much more condensed. So this might be prophase or prometaphase. Okay. So prophase, you see how they can look different, right? So it's like, this would be like the beginning of prophase. This would be like the end of prophase. All right, prometaphase, you're going to see them. Again, this is going to fragment. So you might be able to see it. You might not be able to see it. You might see the centrioles a little bit more. These are going to be more condensed, however. So prometaphase, you'll definitely see the chromosomes. Here we go. So fish blastula again. So here's those centrioles. Here's the spindle fibers. Here it is, lined up, replicated chromosomes lined up in the middle. Here's an up close. So what phase is this? Metaphase. Beautiful. Nice squarish. This is going to be the spindle fibers have lined up. Mitotic spindles have lined up the chromosomes along the metaphase plate or the equatorial plate. And then the chromosomes are, the centrosomes or centrioles, or this is centrioles, are at the poles, and they're going to start elongating the cell and pulling them apart. Anaphase, this is where they're going to pull apart. So if you go back here, this is where that checkpoint is. That M checkpoint is between this, where they've lined up, and this, where they're pulling apart. If they've pulled apart, they've heard the go signal from that checkpoint of yes, they're all attached correctly. Go ahead and pull apart. And they're going to move to the opposite ends of the poles because what's going to happen is in this middle, if it's an animal cell, it's going to form a cleavage furrow. If it's a plant cell, it's going to form a cell plate. Start to form it at the end of telophase. And then at telophase, it's kind of hard to have a picture of because remember, again, for telophase, this is where it's going to reform the nuclear envelope and where those chromatids are going to uncondense into chromatin. So it's a little bit harder to see the chromosomes again. So you're kind of like, if you're too far in telophase, it looks a lot like you're just looking at like interphase of two cells versus where they're just undergoing like pulling apart. So it's a little bit harder to find. If you can find it where the cleavage furrow or the cell plate is just beginning to form, it's much easier to identify. So again, remember mitosis, that prophase, prometaphase, metaphase, anaphase, telophase, that's all just the movement of the chromosomes, the splitting of the nucleus. Once you reform that nucleus, you're then going to split the cytoplasm. That's cytokinesis. And everything in here is the same as everything in here. So you've already replicated all the organelles. You've already replicated everything else. So cytokinesis. So the Golgi is going to send a bunch of vesicles full of cellulose if you're a plant to the middle, and they're going to form up, and they're going to basically form that cell plate, which is that new cell wall in between the two plant cells. And that's what it looks like. So what is this? Cytokinesis. Yep, this is going to be cytokinesis of an animal cell or a cleavage furrow that's forming. Two daughter cells are forming. Two daughter cells with identical copies of the DNA from the parent cell. That's very important for mitosis because what we'll learn after winter break is meiosis, which is a process in which the daughter cells do not have identical copies of the DNA. The cleavage furrow is going to be made up of these units of actin and myosin fibers as it pulls apart. Actin and myosin are the same fibers that make up your skeletal muscle. So they're going to contract and pinch in the middle. So it kind of pinches in and then pulls apart. So normally what we would do is we would do an onion root tip or a fish blastula lab. You would look at a bunch of slides under a microscope and you would have to identify what cells are in what phase and then do a count. So it's kind of the microscope lab basically that we would do before COVID's a thing. So you'd have a lot more microscope time. And then this is a checkpoint for you of checking understanding. So mitosis, it's being able to tell the story of what's happening of these two cells in their process of splitting and identifying all the numbered parts. So identifying that like number one, these are going to be the centrioles. Those are going to be responsible for creating the spindle fibers when they're going to migrate to the poles, when they start to appear. That right here, you have a nuclear envelope. You can't see the DNA. It hasn't actually like split yet. So I would say that Roman numeral 1 is in what phase? If you can't see the DNA and the nuclear envelope is still
interface.
Interphase. Perfect. And so then you start to see these replicated chromosomes, nuclear envelope starts to disappear, spindle fibers are starting to migrate toward the poles. Sorry, centrioles are starting to migrate toward the poles and produce spindle fibers. You can see like number 6 right here, spindle fibers. So it's going through here and identifying, because I'm not going to identify all of them, but go through here and identify some of the parts, all of the parts, all the different regions. understanding what phase you're in. Like for Roman numeral 7, all of these are technically in the same phase. Cytokinesis, by the way, is not part of mitosis. It happens after mitosis because it's the splitting of the entire cell, not the splitting of the nucleus. All right, and we will come back to that, but I wanted to make sure to get to the video that I wanted to share. Let me see. Shoop, shoop, shoop.
To keep DNA organized and regulate access to the genetic code, it is wrapped around structural proteins to form nucleosomes, which are then further organized into fibers called chromatin. The chromatin fibers are packaged into gigantic molecular structures called chromosomes. We are now pulling away from a chromosome, out through a nuclear pore, which is a gateway through the nuclear membrane. We are zooming back to reveal a cell about to undergo mitosis. The nuclear membrane breaks down, releasing the chromosomes. The chromosomes are arranged in the middle of the cell.
See that push me, pull you movement?
Before being pulled apart, with identical sets of DNA going to each side.
That's real time, by the way.
The cell then splits down the middle, creating two new daughter cells.
Oh, this is my favorite part. All right, here's the kinetochore.
This visualization isolates a single chromosome at mitosis. The chromosome contains two identical copies of DNA. Each DNA molecule is packaged up into one of the sausage-shaped chromatids. Sticking out from either side are microtubule fibers from the mitotic spindle used for orienting and guiding the chromosomes to their correct positions. The red region is the kinetochore, which works as the interface between the microtubules and the chromosome. The kinetochore is one of the largest and most complex molecular mechanisms inside living cells, with multiple functions to perform. The kinetochore is central to the movement of the chromosomes and is able to hang on to the dynamic ends of the microtubules. It is also a mechanical tension sensing system, which is able to feel if everything is correctly attached and positioned. It is also a stop signal broadcasting system, sending out chemical signals to the rest of the cell about whether it is ready to undergo separation of the chromosomes. This kinetochore has sensed that the tension is just right and the chromosome is correctly positioned and attached. The last little bit remaining red is the stop signal broadcasting system. It is carried away by a dynein motor, walking down the microtubule away from the kinetochore. These are molecular motors involved with a directed transport along the mitotic spindle. The orange proteins walking to the left are kinesins, molecular motors carrying proteins on their back. Striding from the kinetochore on long molecular legs are dynein, carrying the stop signal broadcasting system. When all the kinetochores are ready, the cell transitions into the anaphase stage, with the chromatids pulled apart to the opposite poles of the cell.
There you go. Well, they're in the remember they're in the nucleus, so this happens pretty quickly because the nuclear envelope's dissolved. Even when they're fortified, they're still in the nuclear envelope? No, because the nuclear envelope's it's dissolved, so that's why it's like it's got to be a pretty quick process. So then, what about all like the enzymes and stuff? They're not really going to interact with them too much, yeah, because it's so densely packed, and by that time, like the spindle fibers have attached. Yeah. You're also careful to concentrate all of your living enzymes and peroxisomes and lysosomes away from. Away, yeah. Your peroxisomes, your lysosomes are very far away. Yeah, yeah. To clarify, the spindle fibers then are just pathways for those sort of blocker proteins and things like that to come in really. For the dimers, yes. Yep. Yeah. So they're not actually doing the pulling. They're just. Well, they do. So the centrioles do pull the spindle fibers a little bit in this like push me, pull you. And that's through polar interactions. And so they do create a tension, and you kind of saw it where it was like it would turn a little teal when it went this way and a little teal when it went that way until they kind of meet that middle. And then when the tension is pretty much the same on either side of the centromere, so in both of those kinetochore regions, that's when the dimer is going to sense that it needs to walk the stop signal away. So it's not actually given a go signal, it's given it is time to remove the stop signal. And that's how your cell cycle works, is basically you go through G1 and there's a stop signal. And it's like, okay, we now have enough nutrients, we can overcome that stop signal. So remove the stop signal and we'll go ahead and go through S phase. And then you go through S and G2, and then there's a stop signal. And then you're like, okay, before we break down our nuclear envelope, we need to make sure all of our DNA is replicated correctly. So that stop signal's there. And then once it says, okay, yes, our DNA is replicated correctly, we can remove that stop signal. And so then it removes the stop signal and it goes through mitosis until it gets to like in between metaphase, anaphase. And then that's what you just saw, is the kinetochores reaching that tension. And then when it says that it's reached that point, they're in the middle. That's the stop signal that gets walked away. And then it can go ahead and go. So if you genetically have genes, which we call oncogenes, which are genes that promote cancer growth, if you have genes that remove those stop signals, your cell will not stop at that checkpoint. So it will basically breeze through that checkpoint, even if the DNA is incorrectly replicated, which is how you end up with mutations and you end up with cancer growth. Yeah. So you can have genetically, you can have genes that you inherit that give you cancer. So one of the most, not all cancers are heritable. One of the most heritable cancer is going to be colon cancer. Yeah. So you can have a mutation that basically turns on certain genes and turns off other genes, and eventually in your life, colon cancer will result. Which is why older people always get the joy of colonoscopies. Yes, which is it's basically a preventative test for colon cancer. It's a sweep. Because it's also really preventable and treatable if you can catch it on time.
There's a wonderful Peter Yarrow song about colonoscopy.
Oh, is there?
Yeah.
Oh,
he wrote CBS did this like PSA and they had him write this song. It's really funny.
I love it. That's awesome. They're horrible. I mean, colonoscopies are just, I mean, they're not, there's a purpose to a colonoscopy, but it's not a procedure that I would say, yay, I have a colonoscopy today. Like that's not, no. But everyone will get one eventually in your life. Well, hopefully. You want to scan for that. But if you're curious, if you want to watch that video, by the way, chromosome kinetochore animation, it's 4 minutes long. It's there. It's the bottom of the page. And then there's discussion on cell cycle, cancer, oncogenes. BRCA1 and BRCA2 are gene mutations that are responsible for your cells breezing through the checkpoints for breast cancer. So BRCA1 and BRCA2 are responsible for breast cancer. And then HeLa cells, we won't get into much this year because we kind of had to cut it. But if you're curious, it stands for Henrietta Lacks. She had cervical cancer. And I'm going to click on this really quickly because I like to talk about it. There's a lot more I could go into about medical ethics that we have because of her. But essentially, her cells were taken without her permission to use them for research. And that's not something that would hopefully happen nowadays because a hospital has to ask you. You have to sign off on it when they remove something from you that you're okay if it's used for research or for studying. And if you're not okay, then they dispose of it. But the hospital just took her cells. And these are the cells that we call an immortal line. So for years and years before this, scientists were working on finding cells that they could keep alive in petri dishes in order to study genetics and disease and how cell growth works and different cellular mechanisms. And her cells actually were able to live in a petri dish in a nutrient agar. And so we call them it's an immortal line. So her cells are able to just be propagated in a test tube. So these HeLa cells, yep. So it's a whole it's a whole article on her if you're curious. But here we go. Yeah, all HeLa cells strung together could wrap around the earth 3 times. Together, her cells would weigh 50 million metric tons. There are more than 60,000 research articles using HeLa cells are published, and a batch of HeLa cells from the tissue culture facility can be purchased for $11. You do, however, have to have a lot of specific safety protocols when doing research with HeLa cells because they are so very prolific. They can get into samples of other cells and then take them over. Yeah. So they'll grow over other cells, but she had cervical cancer. So the fact that we have the HPV vaccine is because of HeLa cells. We also have a lot of other vaccines because of HeLa cells. And what makes it this ethical quandary is that she was never told that her cells were used. Her cells are used around the world for countless research, and her family is compensated in no way. So that's kind of this. And then some shenanigans went on later when they needed to compare her DNA because these are from a tumor that they removed from her. And it had mutated DNA, but they wanted like the source DNA. So they went back to her family and they took blood samples, but they just told the family that it was because they needed to compare it to hers to make sure they didn't have the same disease. So they essentially lied to the family in order to collect their blood and their DNA. Yeah. Yeah. So. Yeah, no, these are her cells. Yeah, they're named after her. So it's Henrietta Lacks. It's the first 2 initials of her first name and the first 2 initials of her last name. Yeah. No. They literally, it's from her tumor. They took her tumor and then propagated cells from it. No, it'll never die. They just propagate super easily in a petri dish. Yeah. So her cells are way older than she would ever be. They're very cool. All right. That is it. Class is over. It is lunchtime. So if you are a digital learner, go enjoy some lunch. Yay. If you have any questions, you can ask. Or I do have a meeting in the afternoon, but it doesn't start till 3:15. So I do have office hours from 2:50 to 3:15 if you have any questions. If you're Herndon and you want to come to my office hours, you're welcome. It's the same link that you just clicked on to get into class. So now you have the link. Welcome. All right. Other than that, adios, arrivederci, au revoir. We're doing the click and learn on Thursday. Bye, guys.
Thank you.
Thank you.
Thank you.
Thank you.
Oh, and junior class meeting today. Yeah, it's a junior class Zoom meeting. So you don't have to go to your advisory. You just click on the Zoom that Dr. Wilson sent you.